Bile is a greenish liquid that's made by the liver and is stored in the gallbladder. Bile's a bit like an alkaline soup, and its ingredients include a variety of organic molecules. Bile does a number of things, including helping with digestion, absorption of fats, and excretion of various molecules. Normally, lipids are insoluble in water, so that's why bile is needed to help emulsify and solubilize them. The organic composition of bile is mainly made up of bile salts and phospholipids, with cholesterol and bile pigments, called bilirubin, making up only a small percentage. Bile is first manufactured in the liver by cells called hepatocytes. Hepatocytes use an enzyme called 7-alpha hydroxylase to convert cholesterol into two primary bile acids, called cholic acid and chenodeoxycholic acid. In the intestines, some of these primary bile acids get dehydroxylated, giving rise to secondary bile acids, deoxycholic acid and lithocholic acid. The liver can conjugate or attach the amino acids glycine or taurine to all four of these bile acids, and this ultimately gives rise to 8 different forms of bile salts. And it's these bile salts that are the main components of bile. So, bile is made in the liver and flows into the intestines. The journey starts when bile flows into the left and right hepatic ducts, which eventually merge to form the common hepatic duct. The common hepatic duct then leads to the cystic duct, which brings the bile to the gallbladder. The gallbladder is a small pear-shaped, hollow organ located beneath the liver, and this is where bile is stored and becomes more concentrated. Approximately 30 minutes after consuming a meal, the food is broken down into a slurry called a chyme, and the chyme begins to enter the first part of the small intestine, the duodenum. When that happens, eye cells, which are in the mucosal lining of the intestine, secrete a hormone called cholecystokinin, or CCK, into the bloodstream. Cholecystokinin travels through the blood and reaches the gallbladder, causing it to contract really strongly. These strong contractions cause the stored bile to get squirted out of the gallbladder and into the common bile duct. The bile ducts are lined by cells called cholangiocytes, which secrete bicarbonate-rich fluid into the biliary tree. Cholecystokinin also causes the relaxation of the sphincter of Oddi, the muscular valve between the common bile duct and the duodenum. This allows the bile and the bicarbonate-rich fluid to flow through the sphincter of Oddi and into the duodenum. Bile salts are amphipathic, meaning that the molecules have parts that are hydrophilic or water-loving and other parts that are hydrophobic or water-fearing. This unique structure allows bile salts to self-assemble into mixed micelles when they enter the intestines. Micelles have a hydrophilic exterior and a hydrophobic interior, and little droplets of fat from the diet can then settle within this hydrophobic interior. This is how bile salts emulsify or break down large fat globules into small emulsion droplets. If you compare a large fat globule to many small ones, the biggest difference is surface area. In other words, by emulsifying the fat, micelles help generate a much greater surface area for the enzyme pancreatic lipase to digest lipids into fatty acids and monoglycerides. Micelles move lipids to the brush border of the intestinal cells where they diffuse out and are absorbed through the intestinal villi. Without the help of bile salts, lipids wouldn't get digested and absorbed, and they'd get excreted in the feces, causing steatorrhea, an oily type of stool. In addition to lipid digestion and absorption, bile acids also help with bilirubin excretion. Bilirubin is the pigment that gives bile its color, and it's actually a byproduct of hemoglobin metabolism. As red blood cells near the end of their lifespan, which is about 120 days, they're destroyed by immune cells like macrophages. Macrophages break down hemoglobin, which are iron-rich proteins inside blood cells that bind oxygen into heme and iron. Next, an enzyme called heme oxidase converts heme to biliverdin, which is green. Then, the enzyme biliverdin reductase converts biliverdin into bilirubin, which is yellow. Finally, the bilirubin leaves the macrophage and enters the blood. The bilirubin leaving the macrophage is not water-soluble and is therefore referred to as indirect or unconjugated bilirubin. Albumin in the blood binds to the unconjugated bilirubin in order to give it a lift over to the liver, where it's taken up by hepatocytes. Within the hepatocytes, the enzyme uridine glucuronil transferase conjugates the unconjugated bilirubin to glucuronic acid, turning it into conjugated bilirubin. And the conjugation of bilirubin allows it to become more soluble. As a result, conjugated bilirubin can be secreted into bile, whereas unconjugated bilirubin is typically not found in bile. So ultimately, bile containing the conjugated bilirubin flows into the intestines. In the intestines, certain bacteria hydrolyze and reduce conjugated bilirubin into a colorless compound called urobilinogen, which can enter various pathways. Most of the urobilinogen, also called stercobilinogen when it's in the intestines, becomes oxidized by gut bacteria to form stercobilin. Stercobilin is what makes feces brown in color. Now, there's a system by which both bilirubin and bile acids get recycled, called enterohepatic circulation. A small amount of urobilinogen that's not excreted in the feces gets reabsorbed from the gut into the bloodstream. Some of that urobilinogen travels through the blood to the kidneys, where it's converted to urobilin, which is very water-soluble and therefore excreted into the urine and gives urine its yellow color. The rest goes right back to the liver to get sent out into the intestines once again. Now, unlike bilirubin, where only a little bit is recycled through enterohepatic circulation, it turns out that roughly 95% of the bile acids get recycled that way, and only about 5% are lost in feces. In fact, there are sodium bile salt cotransporters within the intestine which specifically bring bile salts back into the enterocytes. These bile acids get into the blood and ultimately back into the liver so that they can get added back into the bile once again. Alright, as a quick recap. Bile is formed by the liver, stored in the gallbladder, and released into the duodenum. Bile salts are able to emulsify large lipid globules into smaller droplets to create a greater surface area upon which pancreatic lipase is able to work. Aside from bile salts, another component of bile is a pigment known as bilirubin, which is a byproduct of red blood cell degradation. Both bile salts and bilirubin can be recycled via enterohepatic circulation, where they're transported from the intestines back to the liver.
Carbohydrates include both simple sugars, which are little ring-shaped molecules made of carbon, hydrogen, and oxygen, either alone or in pairs, as well as more complex carbohydrates, which are formed when these rings link up together to make long chains. Carbohydrates provide us with calories, or energy, and simple sugars in particular play a lot of roles in our diet. They sweeten lemonade, balance out an acidic miso soup, fuel yeast in rising dough and alcohol, and help preserve jams and jellies. Now, sugars are found naturally in plants like fruits, vegetables, and grains, as well as animal products like milk and cheese. Added sugars are the sugars that get added to foods, like cereals, ketchup, energy bars, and even salad dressings. To be clear, even if the sugar being added comes from a natural source like sugar cane or honey, it's still considered an added sugar. In fact, a variety of ingredients listed on food labels might be sources of added sugars, some of which you're probably familiar with. Sugar actually refers to a family of molecules called saccharides. Monosaccharides, where mono means one, so one sugar molecule. Disaccharides, where di means two, so two sugar molecules linked together. Oligosaccharides, where oligo means a few, so it's 3 to 9 sugar molecules linked together. And polysaccharides, where poly means many, so it's 10 or more sugar molecules linked together. Glucose is the most important member of the sugar family, and it's a monosaccharide. It's one of the main sources of calories for the body, and is able to cross the blood-brain barrier and nourish the brain. Another monosaccharide is fructose, which is commonly found in honey, fruits, and root vegetables. Finally, there's the monosaccharide galactose, known as milk sugar. It's known as milk sugar because it's only found in nature when it links with glucose to form lactose, a disaccharide found in the milk of mammals, which includes cow milk as well as human breast milk. Sucrose, or table sugar, is another disaccharide, and it's formed when fructose links up with glucose. Sucrose is found in various fruits and vegetables, with sugar cane and sugar beets having the highest quantities. Maltose is another disaccharide, and this one is two glucose molecules linked together, and it's found in molasses, which can be used as a substrate to ferment beer. Sugars, like fructose for example, are most always found in combination with other sugars, and the combinations can be pretty different, even in seemingly similar foods. For example, in honey, 50% of the sugar is fructose, 44% is glucose, 4% is galactose, and 2% is maltose. Whereas in maple syrup, less than 1% of the sugar is fructose, 3% is glucose, and 96% is sucrose. So simple sugars, whether they're natural or added, are mixtures of monosaccharides or disaccharides. Next, there are the complex carbohydrates. There are oligosaccharides, like galacto-oligosaccharides, which are short chains of galactose molecules, like those found in soybeans. Then there are polysaccharides, which are even larger chains with branches, and are the most abundant type of carbohydrates found in food. Starches are polysaccharides with molecular bonds between sugar molecules that human intestinal enzymes can break down. Starches are an important source of calories, and can be found in foods like rice, potatoes, wheat, and maize. Starches don't taste sweet like simple sugars, because they don't activate the taste buds in the same way. And there are also dietary fibers, which are carbohydrates that intestinal enzymes can't break down, and so the body can't digest them. Now, there are many different types of dietary fibers, and they're not all the same when it comes to their structure or impact on health. Fibers have molecular bonds that are resistant to human intestinal enzymes, so they pass through the small intestine undigested, get broken down a bit by bacteria in the large intestine, and ultimately end up as bulk matter in the stool. Fibers are critical because they can slow down the rate of absorption of simple sugars like glucose in the small intestine, which can help maintain healthy blood glucose levels. They also increase stool weight, which helps prevent constipation. And fibers like beta-glucan are also good for heart health. Monosaccharides link together through glycosidic bonding, which is when an OH group from the carbon on one monosaccharide binds with the hydrogen from the carbon of another monosaccharide. Together this forms an H2O, or a water molecule, which goes away. In the case of maltose, that leaves an alpha-1,4 glycosidic bond, which is a bond between carbon number 1 of one glucose monosaccharide and carbon number 4 of the other glucose monosaccharide. And alpha refers to the fact that the molecules are lined up next to each other. Lactose, on the other hand, has a beta-1,4 glycosidic bond, meaning that carbon 1 of galactose and carbon 4 of glucose are bonded, but this time the molecules are stacked with one higher than the other. Finally, sucrose has an alpha-1,2 glycosidic bond, meaning that carbon 1 of glucose and carbon 2 of fructose are bonded. Now, when you eat something, like a piece of onion bread, Enzymes start breaking down the disaccharides, oligosaccharides, and polysaccharides into monosaccharides so they can be absorbed. Different enzymes help to break down different linkages. For example, amylases break down large polysaccharides like starch into smaller units, whereas lactase, sucrase, and maltase break down lactose, sucrose, and maltose into their monosaccharides. The individual monosaccharides that result from the digestion of larger carbohydrate molecules, glucose, fructose, and galactose, cross the gut lining and get into the bloodstream to get used by the body. When glucose levels in the blood increase after eating, the pancreas releases the hormone insulin, and this helps move glucose into all cells for energy. Insulin helps stimulate the liver to store glucose as glycogen in a process called glycogenesis, which is when some of the glucose molecules get linked together with alpha-1,4 and alpha-1,6 glycosidic bonds to form a polysaccharide called glycogen. Insulin also promotes fat and protein synthesis. Now, metabolism of galactose has an initial step where an enzyme in the liver converts galactose into glucose. It basically flips the orientation of the OH group on the fourth carbon. Just like that, galactose can become just another glucose molecule in the liver. Fructose, though, is handled a bit differently by the liver. Fructose has a total of 6 carbons, and most of it's broken down into 2 3-carbon molecules and sent into glycolysis to help generate energy. When energy is needed, all 3 monosaccharides are metabolized through glycolysis, then the citric acid cycle, and oxidative phosphorylation. Ultimately, all digestible carbohydrates, regardless of whether they come from simple sugars in honey or from starches in baked potatoes, are broken down into their component monosaccharides for immediate energy use or stored away for a rainy day, depending on what the body needs. The National Academies of Sciences, Engineering, and Medicine recommended that a healthy diet contain 45 to 65% of its calories from carbohydrates. The number of calories you need to maintain your weight depends on things like your age, sex, height, weight, and activity level. For example, let's take this slightly active 40-year-old woman who's 5 foot 9 inches and 160 pounds, with a BMI of 23.6 and requires a 2,000 calorie diet. And let's say she wants to aim for 55% of her calories from carbohydrates. So that's 55% of 2,000 or 1,100 calories. And there are different types of carbohydrates. First, there's fiber. The general recommendation is to get 28 grams of fiber in a 2,000 calorie diet. With roughly 0 to 2 calories per gram of fiber, that's about 56 calories, or about 3% of her total calories. Next, there's sugars, both the kind that are added to foods and those naturally found in whole foods. There are few formal recommendations for total or natural sugar intake. However, updated Canadian nutrition labels are based on a daily value of 100 grams or 400 calories from total sugars, which is 20% of a 2,000 calorie diet. When it comes to added sugars, both the World Health Organization and the US Dietary Guidelines recommend that they make up fewer than 10% of total calories. Just like her goal for total carbohydrates, she's aiming for the mid-range of the added sugars recommendation, which would be 100 calories or 5% of her total calories. Using this approach, the remainder of her total sugar calories, 300 calories or 75 grams, would come from the sugars found in fruits, vegetables, dairy, and grains. This would be 15% of her total calories, and that leaves 640 calories or 160 grams from starches, or 32% of her total calories, to reach her carbohydrate intake goal. Now, eating a healthy diet means choosing foods that are as nutrient-rich as possible, and foods that contain fiber, starch, and natural sugars like fruits and vegetables tend to be richer in nutrients than those with added sugars. Having said that, processed and packaged foods are a part of most people's diets, so carefully reading nutrition labels can help you compare foods and choose more nutrient-rich options. Generally speaking, picking foods and beverages that are higher in nutrients like fiber and lower in added sugars is best. Alright, as a quick recap, there are various types of carbohydrates. Simple sugars are monosaccharides and disaccharides that the body can readily absorb. Starches are polysaccharides that take longer to absorb. And fibers are polysaccharides that the body can only partially absorb with the help of gut bacteria. Ultimately, a healthy diet includes all types of carbohydrates, coming from a variety of sources like fruits, vegetables, dairy, and grains. It can include added sugars too, with the World Health Organization and the US dietary guidelines recommending that added sugars make up less than 10% of your overall calories. Some people eat to live, and others live to eat. For both groups, the first step of digestion is chewing, or mastication. This is where food gets moistened by saliva and is broken down into smaller bits that are easy to swallow and pass through the esophagus. It's also the step that helps smear the food out over the tongue so that it can be fully tasted. The journey of food starts in the oral cavity, which is the first part of the digestive tract. The oral cavity is like an empty room. There's the roof, which is formed by the hard and soft palate, the floor, which is formed by the tongue and the mylohyoid muscles, the lateral walls formed by the inside of the cheeks, and then there's the front, which gets sealed off by the lips and teeth. A layer of epithelial cells line the inside of the mouth and form the first line of defense against pathogens. The surface of the epithelial cells is kept moist by mucus secreted by salivary glands. The major salivary glands are actually located outside the oral cavity. These are the parotid glands found in front of each ear, the submandibular or submaxillary glands found under the mandible, and the sublingual glands that sit beneath the tongue, under the floor of the mouth. Now, even though the salivary glands are not located inside the mouth, they have ducts that travel to the oral cavity so they can secrete saliva into it. The secretion of saliva is mainly controlled by the parasympathetic nervous system, through cranial nerves. The parotid glands are innervated by the glossopharyngeal nerve, or cranial nerve 9, while the submandibular and sublingual glands are innervated by the facial nerve, or cranial nerve 7. Saliva is mostly made of water, but it has other important components like salivary amylase, which is a digestive enzyme that breaks down starch, and mineral salts like sodium bicarbonate that help maintain a pH of 6.5 to 7.5 inside the mouth. It also contains mucus, which protects the oral mucosa from drying out from the air that blows by when we speak or whistle. Alright, so now when you take that first delicious bite of spaghetti with pesto sauce, you first have to chew the food so it's easy to swallow. This is called mastication, and it involves moving the jaw at the temporomandibular joint. This is coordinated by numerous muscles, including the temporalis muscle, which is fan-shaped and on both sides of the cranium, the masseter muscle, which connects to the mandible and the zygomatic arch of the temporal bone, the medial pterygoid muscle, which connects to the mandible and medial pterygoid plate, and the lateral pterygoid muscle, located at the condylar process. These muscles are innervated by branches of the trigeminal nerve, or cranial nerve 5, and they work together to move the jaw up and down, allowing the teeth to grind and mechanically break down food. The tongue also comes in to help. It moves from side to side, repositioning food and pushing it between the teeth for extra grinding. Now, as we chew, the food gets detected by mechanoreceptors near the periodontal ligaments around the roots of each tooth, as well as by taste receptors on the tongue. These receptors send information through the trigeminal, facial, and glossopharyngeal nerves to a group of salivatory nuclei in the brainstem. The salivatory nuclei then send efferent information through the facial and glossopharyngeal nerves to the salivary glands, which secrete more saliva. Now, it turns out that just the sight or smell of that spaghetti with pesto sauce can also cause the secretion of saliva, and it's based on learned behavior. In fact, this goes back to observations from the famous researcher Ivan Pavlov. who observed that his dog would start drooling at the sight of food. Pavlov then trained his dog to associate food with a bell, and found that eventually the dog would drool whenever it heard the bell. This is called classical conditioning. And now that you've seen this, you might crave spaghetti with pesto sauce when you hear this sound. Alright, now when saliva mixes with food inside the mouth, the digestive enzyme amylase starts breaking down carbohydrates found in foods like potatoes and rice. And when the food is chewed and mixed with saliva, it creates a soft, mushy mass called a bolus, which is ready to be swallowed. The saliva also lubricates the surfaces of the mouth and pharynx, making it easier for the food to go down. So eventually the spaghetti with pesto sauce gets swallowed, which is also called deglutition. And the food bolus moves from the mouth to the oropharynx, or the throat. Now, the pharynx is divided into 3 parts: the nasopharynx, oropharynx, and laryngopharynx. And swallowing is coordinated by muscles under both voluntary and involuntary control. And it can be broken down into 3 phases: the oral phase, the pharyngeal phase, and the esophageal phase. The oral phase starts with the voluntary movement of the tongue. So when the food is ready to be swallowed, the tip of the tongue lifts up and the base of the tongue moves backwards, pushing the bolus to the back of the mouth and oropharynx. This initiates the involuntary pharyngeal phase, where the food bolus passes through the pharynx and into the esophagus. During this phase, mechanoreceptors, thermoreceptors, and taste receptors in the pharynx and upper esophagus detect the food. These receptors send afferent sensory information through the trigeminal, glossopharyngeal, and vagus nerves to the medulla, which is where a region called the swallowing center is located that controls the involuntary phases of swallowing. The swallowing center sends efferent motor information mainly through the glossopharyngeal and vagus nerves to the muscles of the pharynx and upper esophagus. The pharyngeal phase of swallowing starts when this efferent information causes the soft palate and uvula to move upwards, closing the path to the nasopharynx so food cannot reflux up into the nasal cavity. At the same time, it makes the epiglottis, which is a flap-like structure, move downwards and cover the larynx to prevent food from entering the trachea and respiratory tract. Finally, the upper esophageal sphincter, which is a muscular ring that guards the entrance to the esophagus, relaxes, allowing the food bolus to slip through. The epiglottis then moves back to its original position to restore breathing. In the last phase, the esophageal phase, the food bolus passes into the esophagus and the upper esophageal sphincter closes to prevent food from refluxing back into the pharynx. Then the swallowing center sends a message through the vagus nerve to stimulate an involuntary muscular movement of the esophagus, called peristalsis. Peristalsis is a wave-like motion that passes through the esophagus and squeezes the bolus downwards, to the lower esophageal sphincter, which is the entrance to the stomach. Finally, the lower esophageal sphincter relaxes and allows the food bolus to enter inside the stomach. With solid food, swallowing takes about 10 seconds to pass from the mouth to the stomach, whereas with non-solid food, like soup, it only takes about 1 second. Alright, as a quick recap. When we eat, the first step is to break food down by chewing and amylase in the saliva so it's easy to swallow. Then the step of swallowing starts, which includes the voluntary oral phase, where the tongue pushes the food bolus to the pharynx. This triggers the involuntary pharyngeal and esophageal phases, coordinated by the swallowing center in the medulla, which prevent food from refluxing back to the nasal cavity or entering the trachea and lungs. Finally, a peristaltic contraction of the esophagus carries the food bolus downwards and into the stomach. Fats are an essential part of a healthy diet. They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a croissant. Fats are also a major source of energy and a critical component of cells and tissues, and they also help absorb essential vitamins, and can be converted into other molecules like prostaglandins, which help cells communicate with each other. Fats have a 3-carbon backbone called glycerol, as well as fatty acid chains. The fatty acid chain is basically a string of carbon and hydrogen atoms. When an OH group from the glycerol molecule binds to a hydrogen from the fatty acid, an H2O or water molecule gets released. And the two molecules link up. If this happens once, the result is a monoglyceride. If it happens twice, it's a diglyceride, and 3 times makes a triglyceride. Now, there are various types of fatty acid chains, and one way to categorize them is by their length. In other words, how many carbons they have. Short chain fatty acids have 2 to 5 carbons, medium chain fatty acids have 6 to 12 carbons, and long chain fatty acids have 13 or more carbons. Fatty acid chains are also categorized by the bonds connecting the carbons in the chain. A single bond is just one bond between the carbon atoms. And when a fatty acid chain has only single bonds, it's called a saturated fatty acid, because it has as many hydrogen atoms as possible, or it's saturated with them. Triglycerides with saturated fatty acids are nice and straight, so they pack together really well. And as a result, they're usually solid at room temperature. And the longer the saturated fatty acid chain, the more likely it'll be solid at room temperature. Carbons can also have double bonds between them though. And when a fatty acid has one or more double bonds, it's called an unsaturated fatty acid, because it's not saturated with hydrogen atoms. For every double bond, there are 2 fewer hydrogen atoms. Also, a double bond causes a kink in the molecule, so that unsaturated fats don't pack together as nicely as saturated fats. As a result, unsaturated fats are usually liquid at room temperature. Unsaturated fatty acids can be further classified, according to the number of their double bonds. Monounsaturated fatty acids are unsaturated fatty acids with just one double bond. Polyunsaturated fatty acids have 2 or more double bonds. Also, they can be classified according to their location as well. Since all these hydrogens can get kind of crazy looking, we'll just take them away for now. So another name for the methyl end is the omega end. And then we can count the number of carbons until the first double bond. Since this one is 3, it would be an omega-3 fatty acid. If the double bond is 6 carbons from the end, it's an omega-6, and if it's 9 carbons from the end, it's called omega-9. Now, to make things even easier when looking at these molecules, I'm just going to show the bonds. Alright, so omega-3s are usually polyunsaturated fatty acids, and include alpha-linolenic acid, or ALA, eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. EPA and DHA are marine sources of omega-3s. They're produced by microalgae and end up in the tissues of fish like anchovies, mackerel, salmon, and sardines. ALA is found in plants like flaxseed, walnuts, and canola and soybean oils. Our bodies can convert ALA into EPA and DHA, but it's an inefficient process that yields only small quantities. And that's why dietary recommendations include foods that have EPA and DHA. Omega-6 fatty acids are also usually polyunsaturated, and include linoleic acid and arachidonic acid. Linoleic acid is found in oils like safflower, corn, and soybean oils. Arachidonic acid is found in animal sources like fish, meat, and eggs. Our bodies can convert linoleic acid into arachidonic acid, but once again the process is inefficient. Because ALA and linoleic acid can only be obtained in the diet, they're considered essential fatty acids. Omega-9 fatty acids are typically monounsaturated fatty acids, and an example would be oleic acid, and these can be made by the human body. Foods like canola and olive oil, as well as almonds, contain omega-9s. Now, looking at the double bond of this unsaturated fatty acid, like most unsaturated fats, it's got a cis configuration. In a cis configuration, the two functional groups are on the same side of the double bonded carbons. Now, when this happens, the fatty acid chain naturally bends. A molecule that bends does not pack tightly together, so it's a lot more fluid. Think about cooking oils, which are liquid at room temperature. Some fats are in a trans configuration, though. In a trans configuration, the functional groups are on opposite sides of the double bonded carbons, and this keeps the chain more straight and easier to pack. Trans fats result from a process called partial hydrogenation. In just plain old hydrogenation, hydrogens are added to cis fats to get rid of all of the double bonds. So let's say that you've got this triglyceride, and it has a total of 2 double bonds. So in this case, we'd add 4 hydrogens, 2 for each double bond. That turns the unsaturated fatty acids with cis double bonds into saturated fatty acids. Partial hydrogenation, on the other hand, refers to adding hydrogens to most, but not all double bonds. So let's say now that we add just 2 hydrogens. When this happens, some double bonds can be turned into single bonds by the hydrogens, but then they might reform. And what you end up with is an unsaturated fat, but some of those fats have trans double bonds. Partial hydrogenation is a process that happens naturally in the digestive tract of some animals, like cows and pigs, which is why trans fats can be found naturally in meat and dairy products. Partial hydrogenation is also sometimes used in the food industry, in the processing of liquid oils to make them more solid, which can actually lead to the creation of trans fats. Partially hydrogenated oils have been largely removed from foods in North America and Europe, because trans fats have been associated with coronary heart disease. Although some foods might have more of one type of fat than another, the truth is that all foods are made up of a blend of fatty acids. When you eat a food like peanut butter, which has about 75% of its calories from fat, the body goes through a set of steps to digest and absorb the fatty acids. First of all, triglycerides are hydrophobic, so they form large globules of fat, like what you see when you pour oil in water. Enzymes called lipases in the saliva, stomach, and secreted by the pancreas can break down triglycerides into free fatty acids and monoglycerides. But working on the surface of a globule is inefficient. So to speed things up, bile salts produced by the liver break the large fat droplet into smaller droplets, which increases the surface area for the lipases to work. Once the triglycerides are broken down into monoglycerides and free fatty acids, these self-assemble into mixed micelles, which have a hydrophobic interior and a hydrophilic or water-loving exterior. The micelles glide through the watery environment of the intestinal lumen and reach the enterocytes in the intestinal wall. When they get to the enterocytes, the micelles release the fatty acids and monoglycerides, which diffuse into the enterocyte. Inside the enterocyte, the fatty acids and monoglycerides reassemble into triglycerides, and these get packed into a larger structure called a chylomicron. The chylomicron has lipids and proteins, so it's a lipoprotein. It has an outer membrane with phospholipids and proteins, and a hydrophobic core that has triglycerides, cholesterol, and fat-soluble vitamins A, D, E, and K. The chylomicron then leaves the enterocyte, but it's too large to get into the endothelial cells, so instead it enters a nearby lymphatic capillary called a lacteal. From there, the chylomicron floats in the lymph and flows into the thoracic duct, and then gets dumped into the blood, essentially bypassing the portal vein. Once in the blood, the chylomicron releases fatty acids and monoglycerides in peripheral tissues like muscle, which use them for energy, as well as adipose tissue which can store them. After delivering the triglycerides, the chylomicron shrinks in size and eventually gets engulfed by the liver. Now, fats play a super important role throughout the body. They have a huge number of health benefits, and those benefits can vary by the type of fat we eat. For example, polyunsaturated fats are precursors for hormone-like molecules called prostaglandins that stimulate endothelial cells that line blood vessels to release nitric oxide. Nitric oxide is a vasodilator, so that decreases resistance to blood flow and in turn lowers blood pressure. Polyunsaturated fatty acids can also help reduce the total and LDL cholesterol, and that's linked to lower rates of cardiovascular diseases like heart attacks and stroke. Long-chain omega-3 fatty acids like DHA and EPA can both help to lower plasma triglyceride levels, which also protects against cardiovascular disease. Finally, DHA is important in the development of eyes and brains of young infants. The impact of saturated fat on cardiovascular health is a little more complex. Generally speaking, it's recommended to keep consumption of saturated fat low, but just like with unsaturated fats, there are different types of saturated fats. Evidence suggests that different types of saturated fatty acids might have different effects on our cardiovascular health. Evidence also suggests that the health impact of reducing or replacing saturated fat in the diet depends on the nutrient that replaces it. For example, replacing saturated fat with polyunsaturated fat has been shown to benefit cardiovascular health, whereas replacing saturated fat with refined carbohydrate has not. Based on this, the National Academies of Medicine recommends consuming 20 to 35% of our daily calories as fat for a 2,000 daily calorie intake. That comes out to between 400 and 700 calories from fat, or about 44 to 78 grams. And it's not just about the amount of fat that we eat. The type also matters. The World Health Organization and the US Dietary Guidelines recommends that less than 10% of daily calories comes from saturated fats, and that trans fat consumption should be kept as low as possible. Alright, as a quick recap, fats are an essential part of our diet and health. Fatty acids are either saturated or unsaturated, and foods with fat always contain a mixture of fatty acid types. Healthy diets emphasize mono and polyunsaturated fats over saturated fats, and trans fats are associated with an increased risk of cardiovascular disease. Improvements to our diet and health can be made by focusing more on the type of fat we eat and less on the amount. The pancreas is a long, skinny gland the length of a dollar bill and is found in the upper abdomen, or the epigastric region, behind the stomach. It has endocrine functions, meaning it secretes hormones into the blood that eventually act upon their target tissues. For example, alpha and beta cells in the pancreas make hormones like insulin and glucagon that are secreted into the bloodstream to help regulate blood sugar levels. However, approximately 90% of the pancreas is dedicated to its exocrine functions. The exocrine pancreas secretes enzymes and fluids that help neutralize and digest food within the intestines. The exocrine pancreas can be divided into lobules, each of which contain lots of functional units called acini. An acinus is a cluster of acinar cells that all work together to make digestive enzymes. In fact, the word acinus means berry, which describes the berry-like appearance of these cell clusters. Each acinus secretes digestive enzymes which flow into small intercalated ducts that are lined by ductal cells. These ductal cells secrete bicarbonate and fluids that make up the liquid portion of pancreatic fluid, and ultimately help to neutralize the acidic stomach contents as they enter the intestines. The intercalated ducts merge together, forming an intralobular duct, which join with other intralobular ducts, and finally drain into the main pancreatic duct. The main pancreatic duct travels through the length of the pancreas and drains into the duodenum. Now let's look at the enzymes made by the acinar cells. The main enzymes include pancreatic amylase, which breaks down carbohydrates, trypsin and chymotrypsin, which break down proteins, and lipase, which breaks down lipids. Now, as it turns out, macromolecules like carbohydrates, proteins, and lipids are also found in the cells of the pancreas. Therefore, in order to protect the pancreas from destroying itself, the acinar cells manufacture inactive forms of the enzymes called proenzymes, or zymogens. These zymogens need to be activated by proteases, which cleave off a polypeptide chain, kind of like pulling the pin on a grenade. For additional security, the zymogens are kept away from sensitive organelles in storage vesicles called zymogen granules, and are packaged with protease inhibitors that prevent enzymes from doing damage if they become prematurely active. To digest a meal, these zymogens are released into the pancreatic duct and delivered to the small intestine where they're activated by the protease trypsin. Trypsin is a pancreatic digestive enzyme that's initially produced as the zymogen trypsinogen. Normally, trypsinogen isn't activated until it's cleaved by the protease enteropeptidase, which is found in the duodenum. Now, two enzymes that don't need activation by the protease trypsin are pancreatic lipase and amylase, both of which are secreted in their active forms. Now, the secretions of the ductal cells are filled with various ions, particularly sodium, potassium, chloride, and bicarbonate. The concentrations of sodium and potassium are about the same as the concentrations in the plasma, whereas the concentrations of chloride and bicarbonate depend on the pancreatic flow rate, which is the volume of secretions per minute. The chloride and bicarbonate concentration is regulated by transport channels on ductal cells. On the apical membrane, which is the side of the ductal cell facing the duct, there's a chloride bicarbonate exchanger. And on the basolateral side of the membrane, or the side facing the extracellular fluid, there are both sodium-potassium ATPases and sodium-hydrogen exchangers. Within the pancreatic ductal cell, the enzyme carbonic anhydrase combines carbon dioxide and water into carbonic acid. Carbonic acid then dissociates into bicarbonate and hydrogen. The hydrogen ion is pumped out of the ductal cell and into the blood, in exchange for a sodium ion via the sodium-hydrogen pump, and a bicarbonate ion is pumped into the pancreatic duct, becoming part of the pancreatic fluid, in exchange for a chloride ion. When food is being digested in the intestines, the concentration of bicarbonate in the pancreatic fluid has to be high to offset the acidity of the stomach acid. Consequently, the chloride concentration in the pancreatic fluid will be low, since the chloride ions are being exchanged for bicarbonate ions. So when the pancreas increases secretion, which increases the flow rate, the bicarbonate concentration increases and the chloride concentration decreases. Now, there are three phases of pancreatic fluid secretion: the cephalic, gastric, and intestinal phases. Let's say you're about to eat a nice, big slice of pizza. In the cephalic phase, the sight and smell of the pizza reaches the brain and causes it to send efferent signals through the vagus nerve to the pancreas. The vagus nerve secretes the neurotransmitter acetylcholine, which stimulates muscarinic receptors on acinar cells and causes them to secrete enzymatic fluid. Next is the gastric phase, which is when the chewed up pizza enters the stomach and makes it distend. Distension is sensed by mechanoreceptors in the stomach wall. This afferent information travels through the vagus nerve to the brain, which sends efferent signals back through the vagus nerve to the pancreas. The result is that the acinar cells produce slightly more fluid. Finally, there's the intestinal phase, which is responsible for over 80% of pancreatic secretions. When chyme, or partly digested food and stomach acid, enters the duodenum in the intestinal phase, proteins and lipids are broken down into various components like amino acids and fatty acids by pancreatic enzymes that get produced in the cephalic and gastric phases. These amino acids and fatty acids stimulate eye cells, which are in the lining of the duodenum and jejunum. Eye cells secrete the hormone cholecystokinin, or CCK, which binds to receptors on the acinar cells of the pancreas and strongly stimulates them to increase secretion of digestive enzymes. Chemoreceptors in the intestinal mucosa can also detect the presence of lipids, carbohydrates, and proteins, as well as decrease the pH in the lumen. These chemoreceptors send afferent information to the brain via the vagus nerve, and efferent information comes back to the pancreas through the vagus nerve to increase pancreatic secretion during the intestinal phase. Similar to the acinar cells, ductal cells also have cholecystokinin and acetylcholine receptors. In addition, ductal cells have receptors for a hormone called secretin, which is released during the intestinal phase. Partially digested food arriving from the stomach is acidic, and when it enters the duodenum, the rise in acidity signals S cells that line the duodenum to release secretin. Secretin acts on secretin receptors on the ductal cells to massively increase secretion of the aqueous portion of the pancreatic fluid, which is high in bicarbonate, and therefore helps to neutralize the acidic chyme. When food eventually reaches the ileum and colon, neuroendocrine cells secrete the hormone peptide YY, which helps inhibit pancreatic secretion. Although the exact mechanism here is not well understood. Alright, as a quick recap. Acinar cells of the pancreas produce enzymes that help digest food, and ductal cells produce aqueous secretions that help neutralize the acidic stomach contents. Acinar cells are mainly stimulated by cholecystokinin and the neurotransmitter acetylcholine, whereas ductal cells are mainly stimulated by secretin. Finally, the concentration of sodium and potassium in the pancreatic secretions is relatively constant, but the concentration of bicarbonate increases when there's a higher demand for acid neutralization.
Protein is an essential part of the human diet. It's found in a variety of foods like eggs, dairy, seafood, legumes, meats, nuts, and seeds. Regardless of the source, the protein that we eat gets broken down and reformed into new proteins in our bodies. These proteins do everything from fighting infections to helping cells divide. You name it, they're doing it. At its simplest, a protein is a chain of amino acids bound to one another by peptide bonds, like a string of beads. These strings get twisted and folded into a final protein shape. When we eat protein, it gets broken down into its individual amino acids. Most amino acids have a central carbon atom bonded to one amino or nitrogen-containing group and one carboxylic acid group. That's why it's called an amino acid. The carbon also has one hydrogen atom and a side chain, which is unique to each amino acid. The exception to this is proline, which has a tiny little ring structure instead. Although there are hundreds of amino acids in nature, humans only use about 20 of them to make basically every type of protein. They include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. That's 20. One way to divide them is by defining which ones our bodies can make and which ones we can't. There are 5 amino acids, alanine, asparagine, aspartic acid, glutamic acid, and serine, that we can get from foods, but we can also make ourselves. These are called non-essential amino acids. Then there are 6 that we call conditionally essential because healthy bodies can make them under normal circumstances: arginine, cysteine, glutamine, glycine, proline, and tyrosine. But we can't make them in cases like starvation or certain inborn errors of metabolism. Finally, there are 9 of them that we can only get from food: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. We call these the essential amino acids. Dietary protein provides the essential amino acids that are needed to make our own proteins, hormones, and other important molecules, a circle of life of sorts. But to do so, we need to break the dietary protein down first, through a process called proteolysis. When we first eat a protein-containing food, proteolysis begins when the food reaches the stomach. First, hydrochloric acid denatures the protein, unfolding it and making the amino acid chain more accessible to enzymatic action. Then, pepsin, which is a protein itself made by gastric chief cells, enters the picture. Pepsin cleaves any available protein into smaller oligopeptide chains, which move into the duodenum, where a second set of digestive enzymes, made by the pancreas, further chop the oligopeptides into tripeptides, dipeptides, and individual amino acids. These can all be taken up to the intestinal cells, where di and tripeptides are then converted into amino acids. Some amino acids remain in these cells and are used to synthesize intestinal enzymes and new cells. But most enter the bloodstream and are transported to other parts of the body. In general, animal-based protein foods like eggs, dairy, seafood, and meat provide all 9 essential amino acids in adequate amounts. Soy foods are unique in that they are plant based and also provide all 9 essential amino acids in adequate amounts. Most other plant foods, including whole grains, legumes, nuts, and seeds, have high amounts of some amino acids and low amounts of others. Hearing this, it might be easy to assume that animal based foods provide more protein than plant based ones. But, as it turns out, a cup of tofu has the same number of grams of protein as 3 ounces of steak, chicken, or fish. And half a cup of lentils has more grams of protein than an egg. And not all plant foods are low in the same amino acids. So, eating a variety of plant based foods can provide all 9 of the essentials. For example, pairing protein sources, like rice and beans, or hummus and pita bread, or oatmeal topped with almond butter. However, in terms of volume, it may be necessary to eat more plant-based foods to get a similar amount of protein and amino acid profile provided by animal-based proteins. Generally speaking, daily protein requirements are based on studies that estimate the minimum amount of protein needed to avoid a progressive nitrogen loss. The World Health Organization guidelines and the U.S. recommended dietary allowance each estimate that daily protein requirements for healthy adults are about 0.8 grams per kilogram of body weight. Protein recommendations per day vary by age as well. Children 1 to 3 years of age are recommended to get 13 grams. For ages 4 to 8, 19 grams are recommended. And between ages 9 and 13, 34 grams are recommended. Whether a person is male or female also impacts protein needs. Females ages 14 and above are recommended to get 46 grams of protein per day. Males age 14 to 18 need slightly more, about 52 grams per day. And males 19 and older are recommended to get 56 grams per day. Some groups, like pregnant and breastfeeding women, as well as athletes, have elevated needs, and older adults may also benefit from eating more protein. Getting the right amount of protein per day can be achieved in multiple ways. For example, 1 serving of Greek yogurt with breakfast, a salad topped with 3 ounces of chicken for lunch, and 3 to 4 ounces of fish at dinner provides about 64 grams of protein. Getting the same amount of protein could also be achieved by eating a cup of tofu scramble and a slice of peanut butter toast for breakfast, a cup of shelled edamame with lunch, and 1 cup of lentils and brown rice at dinner, which also offers about 64 grams of protein in total. Now, the fact is that it's still unclear what an optimal amount of protein is, and the research is ongoing. In clinical settings, there are certain individuals who may be at risk for a protein deficiency, including patients with malnutrition, trauma, and burn injuries, as well as various conditions impacting nutrient absorption like inflammatory bowel disease. These individuals may have increased protein needs compared to the general population. Except for certain circumstances like kidney disease, there usually isn't a health risk associated with eating a lot of protein, because our bodies are able to process it. Alright, as a quick recap. Protein is a cornerstone of the human diet, and a major component of our bodies. There are 5 non-essential, 6 conditionally essential, and 9 essential amino acids. We need to get essential amino acids from our diet. Protein needs vary depending on life cycle stage, level of physical activity, and health status. Some health conditions may put people at risk for protein deficiency or increased protein needs. Everyone, whether omnivorous, vegetarian, or vegan, can get enough protein by eating a variety of foods.